Welcome. My name is Max de Rooij, and I'm a third-year PhD candidate at the Department of Biomedical Engineering at Eindhoven University of Technology in the Netherlands. And I will be presenting my poster titled Personalizable Hybrid Modeling of Postprandial C-peptide, which is work I did together with Natal van Riel and Shauna O'Donovan. Universal differential equations are a modeling framework that integrate mechanistic models with neural networks to model incomplete biological systems. The differential equations that compose the mechanistic modeling part describe the known part of the biological system, while the neural network describes the unknown part, which needs to be derived from data. However, biological data exhibits considerable heterogeneity. For example, if we have different individuals in our dataset, or if we have different experimental conditions in our dataset, which conventional UDEs are unable to accommodate directly. The example that we see here is that in diabetes, glucose and C-peptide responses vary widely between individuals. To be able to use universal differential equations in these cases, we introduce conditional universal differential equations, or CUDEs, which contains a shared neural network structure that describes population-level behavior and individual-specific conditioning parameter that captures personal differences in C-peptide production. This structure allows for simultaneous model discovery and personalization without needing a model for each person or excessive data. The model combines a two-compartment ordinary differential equation model of C-peptide kinetics, which is taken from Van Kouter, and a neural network embedded within the production term with two inputs. We have relative plasma glucose relative to the initial value of glucose and an individual parameter we call beta I. This conditional UDE was trained on 117 individuals with normal glucose tolerance, impaired glucose tolerance, and type 2 diabetes mellitus, using the glucose and C-peptide responses to an oral glucose tolerance test. From figure 3, we can see that the conditional UDE fits C-peptide levels across all glucose tolerance groups, while the universal differential equation, which does not contain the conditional parameter, fails to accurately estimate the C-peptide levels across all of the groups. We also see in figure 3b that the conditional UDE shows quite a large reduction in error compared to the regular universal differential equation for many individuals. We also looked at this beta I parameter that I previously mentioned, and we see that this beta I parameter actually correlates strongly with first phase clamp response, which is measured from a hyperglycemic clamp and is actually the gold standard method for measuring insulin or C-peptide production capacity. So now we have a neural network that accurately describes C-peptide production in response to glucose, and we can kind of interpret this beta I parameter. However, for individuals out of the distribution, this may still be quite a risk because neural networks are known for their poor extrapolation capabilities. To be able to also effectively extrapolate outside of the data that this model was trained and tested on, we used symbolic regression, which enabled the recovery of an interpretable expression of C-peptide production resembling glucose-mediated Michaelis-Menten kinetics, and also showing excellent generalization to unseen data. This can be seen in figure 4 on the bottom right of this screen. Finally, the conditional parameter extension allows for an efficient modeling of inter-individual variability in physiological systems. The conditional parameter in this case becomes an interpretable index of beta cell function validated by gold standard measurements. As an additional note, the conditional UDE is quite data efficient, which shows a high performance with as few as 29 individuals if class balance is maintained. Symbolic regression makes this model transparent and deployable in different situations. There are also numerous applications beyond type 2 diabetes, which include personalized cardiovascular modeling, PKPD modeling in drug development, or infectious disease modeling.